I'm glad to be here today to talk about wastewater treatment in the context of climate change and share with you a little bit about my research on aerobic granular sludge, which is an emerging wastewater treatment biotechnology. I'm going to start with the takeaway message. Aerobic granular sludge may be less affected by climate change impacts and also contribute less to climate change than more traditional treatment methods like activated sludge. In this preview image, you can see granules on the left and activated sludge on the right after 5 minutes in these glasses. And they're very clearly different, and to get to why that matters, I'm going to start with a quick explanation of how wastewater treatment works in general and how it is related to climate change. Then I'm going to tell you about what makes granular sludge different and interesting from a climate perspective. And finally, I'll tell you a little bit about my new research program here at MSU and what I'm trying to accomplish with granular sludge with respect to these other water quality benefits that I allude to. Now, wastewater comes from all the expected places in our homes, offices, schools, and so on. Before we discharge this water back to receiving waters, we want to make sure that we remove organics. These are carbon-based molecules because they will consume oxygen in the receiving waters as they degrade, and that's harmful for aquatic life. And we want to remove nutrients like nitrogen and phosphorus, which can cause an overgrowth of algae and also decrease water quality. Various strains of bacteria perform the biochemical reactions to remove these contaminants, and we just need to provide them the right environment. We build treatment reactors with a range of oxygen conditions so that the right kinds of bacteria will grow. We need combinations of aerobic, anaerobic, and anoxic conditions to remove organics like nitrogen and phosphorus. Now, nitrogen and phosphorus are especially tricky because there are multiple species of microbes involved, and they require multiple oxygen conditions in a specific order to achieve complete removal of nitrogen and phosphorus. What this typically looks like is a series of reactors with different oxygen levels. So the different kinds of bacteria are suspended in the wastewater, as you saw in that first image of activated sludge in the glass, and they carry out their part of the process when they encounter the right conditions. In Bozeman, our treatment plant achieves removal of organics and nutrients by operating a series of these 5 reactors with a recycle of flow between them. So the anaerobic and aerobic reactors are for phosphorus removal. These anoxic and aerobic reactors facilitate nitrogen removal. And carbon sources can be consumed throughout the process, but that especially happens here in the aerobic section. Since there are literally tons of bacteria just floating around in the water and they're doing all the work, we need a clarifier here at the end to concentrate and collect those bacteria so that we can send them back to the beginning of the process to do it again. And then the clean water comes out here at the top. Now, the regulatory requirement for nutrient removal is relatively new in the last few decades, but the basic concept of biological wastewater treatment to remove organics has been around and in practice for more than 100 years. So, what does this have to do with climate change, you may ask? Well, it turns out that climate change impacts wastewater treatment, and wastewater treatment contributes to climate change. Now, we'll start with how climate change influences wastewater treatment. And the earlier version of this image was missing this component, stormwater. And we know that one of the most visible impacts of climate change is variation in weather patterns, where some areas are becoming wetter and some are becoming drier. Additionally, extreme weather events like major storms are increasing in frequency. In many places, especially in older cities in the Northeast or the upper Midwest here in the US, stormwater and wastewater collect in the same sewer pipes. This is called combined sewage. And even under small amounts of rain, like when it's falling on a really large impermeable area like a modern city, can amount to a huge volume of runoff, which overwhelms the wastewater treatment plant, which was never designed to handle that amount of water. And the result is that the excess water, which is essentially diluted raw sewage, is discharged directly to surface water. This already happens, and it's expected to get worse with more intense storms. The increase in extreme storm events due to climate change also increases the risk of flooding in low-lying areas, which is typically where wastewater treatment plants are located, and it may result in infrastructure damage at the plant itself, beyond the impact to the water quality. So in summary, these climate changes mean increasing variability in the quantity and quality of water that's coming into and going out of treatment plants. And this makes it more difficult for treatment plants to achieve the kind of high quality effluent that we expect and that's required by our laws. Now, as I said, the relationship between climate change and wastewater goes both ways. So wastewater treatment produces 3 of the major greenhouse gases in direct emissions: carbon dioxide, methane, and nitrous oxide, or N2O. CO2 and methane are end products of anaerobic and aerobic breakdown of carbon sources. Nitrous oxide is an intermediary in the nitrogen removal process. And in the US, wastewater treatment accounts for something on the order of a couple percent of total greenhouse gas emissions, but a larger fraction of methane and nitrous oxide emissions. I think methane is something on the order of the 16%. And so these two, methane and nitrous oxide, of course, are significantly more potent greenhouse gases than CO2. Wastewater treatment also has indirect impacts, which are related to energy consumption and infrastructure. It also takes a lot of energy to operate a treatment plant, with aeration equipment and pumping consuming most of that energy. Finally, the wastewater infrastructure, transmission pipelines, treatment facilities are spread across the landscape, and they require a lot of raw material and energy to construct and maintain. So now we get to aerobic granular sludge, where instead of floating around in the water in very small flocks, here the bacteria which treat the wastewater form these little balls of biofilm, typically a couple millimeters in size. And the bacteria stick together with a slime that they excrete. It's called extracellular polymeric substance, or EPS. And what is fascinating about granular sludge is that each granule is like a little reactor where all of those oxygen conditions coexist, which means that we can achieve simultaneous removal of organics and nitrogen and phosphorus within each individual little granule. Now, these different zones, the oxygen conditions, are controlled by the dissolved oxygen concentration in the bulk fluid, in the wastewater, in the reactor. And so the dissolved oxygen is consumed as it diffuses into the granules, and that allows for contaminant removal to occur within the granule during just a several hour cycle time. Aerobic granules are typically cultivated in a 3-stage sequencing batch reactor, or SBR. And here in the first phase, in the anaerobic phase, dirty wastewater comes in from the bottom and it pushes the clean treated water from the previous cycle out. When the reactor is filled, the air comes on, and that creates these oxygen gradients in each of the individual granules so that all of the removal processes can occur. When the cycle is complete and the water has reached the appropriate effluent condition, then the air gets turned off and all the granules settle very quickly, just like we saw in those first 2 images of the glasses. The granules settle very quickly, and then the cycle starts over again. Because all the reactions occur within the granules, we don't need separate reactors or recycle streams or clarifiers like in a conventional activated sludge plant like this. This image is a granular sludge system in the Netherlands, which operates under the trade name of Nereda. And this total plant serves 140,000 people. And these 2 Nereda reactors treat 7.6 million gallons of wastewater per day, which is 40% of the total flow of wastewater through this plant. Now, there are on the order of 60 to 70 Nereda facilities around the world on 6 continents. North America is a little bit late to the game, and the first full-scale plant was built in Alabama a couple years ago, and there's another one under construction now in Whitefish, Montana, which is very exciting. Now, as you likely noticed from that last image of the treatment plant in the Netherlands, the granular sludge facility takes up much less space, up to 75% less space. And these plants cost less to build and less to operate, in part because they require significantly less energy. Now recall, we're only aerating part of the time, and there's much less pumping required in these reactors. And these are benefits of granular sludge systems which reduce the indirect climate change impacts of wastewater treatment. In addition, granular sludge systems may suffer less impact from climate change related variations in water quantity and quality than traditional systems. Variable loading here means fluctuations in concentration of organics and nutrients and other contaminants that might come through a treatment plant, say from a combined sewer. Because the bacteria are contained within the slime of the biofilm granule, they have quite a bit more protection than suspended and dispersed bacteria do in activated sludge. So effluent water quality is more stable and it recovers faster from shocks to the system. So even though we know all of these benefits of granular sludge, and there are treatment plants in operation around the world, there's still so much that we don't know about granules. When I originally started my research, I used magnetic resonance, the same technology you would use in the hospital for a brain scan, for example. I used MRI to observe the internal structure of the biofilm granules non-invasively. I was curious about differences between granules grown in the lab using synthetic wastewater and those used in real treatment plants. And there are a few of the magnets that I used for this work. These here on the right are here at MSU, and the 950 on the left is in Utrecht in the Netherlands. And the key point to know here is that magnetic resonance measures signal from hydrogen protons. In this case, that's mostly water. And the signal that we get can tell us about how mobile the water is in the very local environment. Like what structure does it experience? This is the reactor that we sampled in Utrecht. We dipped a bucket in during the aeration phase and we collected some granules. Here you can see the settled granules and the clean water on top. This layer here, these are the floccular sludge like you would see in an activated sludge system. And from this sample, we took a granule in a 5mm sample tube and we put it in the magnet. Now what you see here are MRI images of granules from 3 different sources. All of these images show a cross-section of the granules, and there is water in the sample tube outside of the granules. This on the left is one kind of EPS that was extracted from granules and then re-gelled into a new granule with no bacteria in it. So it's a kind of control granule, and it looks pretty homogeneous, like we would expect, since there's only one kind of thing in that granule. The 2 images on the right show granules from 2 different treatment plants in the Netherlands. This is the one that I showed you the image of earlier. Now this Nereda granule from Utrecht looks quite a bit different than this alginate granule here on the left. There are very dark spots and a variation in the coloring here. That indicates that the water has a different environment in these different places in the granule. This Nereda granule from Hammerwolde looks again different from the other 2 samples. There are bright regions that look like the Utrecht granule, but also really dark regions. In the dark regions, these are where we've already lost the signal from the water within the 5.3 milliseconds it took to create this image. Another way to look at granule structure with MRI is called a T2 relaxation map, and this measurement collects a time series of images as the signal decays, and the color here in each pixel is an indication of how long the signal lasted during the measurement. And as you can see, signal from free water in the tube, this green on the outside here, it lasts longer than the signal coming from within the granule, where you see more shades of blue. The darker blue shades here correspond to more dense cell clusters and complex EPS structures. The very light blue granule on the right is that extracted EPS or Calmera granule, which again looks pretty homogeneous since there's only one kind of biopolymer present, rather than all the variety that are present in real granules. These 2 kinds of internal structures that you see on the left are fairly common, and most granules tend to look more like the one here in the middle. Images like these made me really curious to understand more about what causes these structures and how we might be able to use granules for purposes beyond the removal of organics and nutrients from wastewater. I don't have these answers yet, but I am working on it. As you are probably aware, there are all manner of contaminants and synthetic compounds from a variety of sources in the environment, and many of them end up eventually in wastewater. These compounds, which include pharmaceuticals and forever chemicals like perfluoroalkyl substances or PFAS, are typically poorly removed in conventional wastewater treatment plants. They often don't biodegrade via the available microbes and pathways and timelines that are common in wastewater treatment. So my newest research projects are designed to evaluate if that same biofilm structure that we saw in the MRI images might provide a means to improve removal of pharmaceutical and PFAS compounds from wastewater. there is some preliminary evidence from other studies that this EPS may be able to bind up the contaminants while at the same time protecting the bacteria from much of the negative impact of those contaminants. I will be monitoring the extent to which lab granules can do this, while also removing the conventional organics and nutrients that we see in wastewater. This image here shows one of the 4 SBRs that are operating now in my lab, and the lab granules are doing their jobs. I currently have funding from the National Institutes of Health and Montana INBRE program to look at pharmaceutical removal in granular sludge systems, and from the Montana Water Center and USGS to look into removal of PFAS, these forever chemicals, also from granular sludge in lab granules. So I will end where I started. The biofilm structure of granular sludge makes it less susceptible to some of the impacts of climate change, like variations in water quality and quantity, and granules may also contribute less to climate change because of the very significant energy and resource savings they afford. On top of that, I am out to demonstrate that granular sludge can play a part in removing other contaminants like pharmaceuticals and PFAS from wastewater to protect water quality. I thank you very much for your time, and I would like to acknowledge my funding sources and collaborators throughout this research. Thanks very much.